Hello there guys, this is Macram and today we're gonna tackle about meiosis. Sperm meets egg, which then undergoes multiple cell divisions to make a living organism with trillions of cells all doing their own specialized function. Impressive, but where did the egg and sperm come from? The answer is, as you may have guessed, meiosis. First, it's good to know that there are two types of cell division processes. The simpler one is mitosis, which produces two identical cells with exactly the same genetic information. Okay, so the other one is meiosis. It's much more complicated process, creating not two but four cells with only half the number of chromosomes and crucially all genetically different from each other. Both mitosis and meiosis include the same phases: prophase, metaphase, anaphase, and telophase, except in meiosis they happen twice, so they usually refer to as 1 and 2. The easiest way to remember these phases names is to remember it IPMAT. Look at meiosis in more detail. As always, cellular division starts with a process called DNA replication. This involves making two identical copies of original DNA molecule. The cell ends up temporarily with double the normal number of chromosomes. In prophase, the duplicate chromosomes join up with the pay from the other parents, so the mother bind with the father, forming a group of two chromosomes called homologous chromosomes. What happens next is vital for successful meiosis, as each chromosome is lined up next to its partner P. One chromatid from each side gets entangled with the corresponding chromatid from the other side. This is called crossing over. During this brief period, the two chromatids swap certain sections of DNA. This is called recombination. The sections that they trade correspond to the same location so that each chromatid retains a correct number of genes. Recombination is really important because it creates variety. The new cells are identical to other parents and they also want the other as well. They are new genetic combinations. In fact, that's the whole point of sexual reproduction. It increases genetic variability. Each chromatid is not different, and as each one will end up in a separate gamete, and means each sex cells is genetically different from all others. This explains why brothers and sisters are different despite having the same parents. Only identical twins have the same genetic makeup as they both originate from the exact same egg and sperm. Now, back to meiosis. Next comes up metaphase 1. As the chromosomes align themselves up in the middle of the cell, and in anaphase 1, the spindle fibers pull the chromosomes apart to opposite ends. In during telophase 1 and cytokinesis, the cell pinches apart in the middle and the nuclear membrane reforms around the two new daughter cells. That's the end of meiosis 1. Now, for part 2, we start with our recombined daughter cells each, so with 46 chromosomes, but sperm and egg cells only had 23 chromosomes. So we need to cut these cells in half. Meiosis, half second round of division. The process is exactly the same as before, except that there is no DNA replication. We start straight with prophase 2 with chromatin clumping again to form chromosomes, then align in the middle of the cell during metaphase 2, and chromatids are pulled apart during anaphase 2 by the spindle. Verse telophase and cytokinesis pinch the souls together with four new crandall form. The end of meiosis give us four different six cells, each only with 23 chromosomes, ready for future fertilization. So thank you for watching, guys. I hope that you learned so much. It's a very hard, but it's worth it. Thank you and God bless.